Hello there. I welcome you to the new course of our Bioinformatics series, which is Bioinformatics and R. I named this course after our renowned programming language, which is R. And Introduction to Bioinformatics and R is our first module. So, in this module, we will explore the fundamentals of bioinformatics and R programming language. So, let's get started. First of all, I want to discuss what bioinformatics really is. So, in our series, we have made a lot of courses, and now we are mainly focusing on our R course. So, bioinformatics is a field that combines biology and computational analysis. So, it's play a crucial role in various areas of biology, such as genomics, proteomics, and much more. Bioinformatics also help us make sense of complex biological data, like the data of patients in a hospital, in which we have the blood samples data and more of that, like the FPKM values, TPKM values, and stuff like that. So we're gonna gain insight into our biological processing using the technique which is called bioinformatics. So mainly where bioinformatics is being used. So bioinformatics is essential for analyzing and interpreting vast amounts of biological data. It integrates computational method with experimental approaches to uncover patterns, identify genes, and predict protein structures and much more. So it's a powerful tool for advancing biological research. So now we have discussed bioinformatics in biological research, what this main course is about, and now let's talk about R. What is R? You can say why R, what R, but what basically is R programming language? R is a programming language which is quite popular in bioinformatics field. It offers a rich set of tools for the data analysis, statistical modeling, and visualization. Its versatility and flexibility make it an ideal choice for bioinformatics research and analysis. So, what is the main use of R? Basically, R is a platform independent that runs on Mac OS, Linux, and Windows operating system. So, it has an excellent user and developer support as well. So, you can use it on your vast online community. But being as a researcher, you have to deal with a lot of bioinformatics data. So you have to integrate these bioinformatics tools with your websites, databases, and stuff like that. So you have to use R for making some shiny apps and stuff like that. So R has a large and growing collection of statistical, graphical, and data manipulation functions. It has an intuitive syntax and interactive programming environment that makes it easy to learn and use. So our ecosystem is also available on the comprehensive R archive network, which is also called CRAN, that provides a wide range of packages for statistical analysis and data manipulation. Additionally, in our bioinformatics field, bioconductor is a term that is mainly used. So you have heard a lot of bioinformatics techniques that is often combined with bioconductor. So Bioconductor offers specialized packages for bioinformatics analysis such as genomics, proteomics, and transcriptomics. So now we are talking about the R ecosystem. So let's talk about other relevant packages that are available. So there are many other useful packages that are also used for the bioinformatics, which is ddplot, tidy, ggplot, and a lot of other tools like biostrings. So what are the benefits of using R in bioinformatics? So we have seen that it provides a good support, and the team is working on a lot of data that you can use. But what basically is the main importance of using R in your bioinformatics? So R provides a wealth of tools and function for analyzing complex biological data. It has powerful statistical and machine learning algorithm for data mining and modeling. Also, R has data visualization options. So R has advanced data visualization option capabilities like including the ggplot lattice package, which allow compiling and interactive graphs and chart. Also, R has a rich community and supportive community, which also helps the developers and through blogs and social media and YouTube channel as well. So there is a also demo that is included. I'm gonna make a tutorial video on that as well, how to install R. So in the next lecture we are going to set up our R environment. So get started with R in programming. You need to download and install the R software and then integrate that R Studio, which is integrative development environment for the R, which is also user friendly and interface provides a friendlier interface for coding, debugging and mapping projects. So configuring R package. So once we have installed the R and R Studio, you have to configure some packages and libraries that include tidyverse, biostrings and many others. So there are some recommended packages that we are going to use in our biological research is bioconductor, genomic range, GWiz, R color brewer and something like that. So that wraps up our session for the introduction of bioinformatics and R. We covered the basics of bioinformatics, the advantages of using R, and how to set up the development environment, which I am also going to make a tutorial video on that. So now it's time to ask you if you guys have any questions and any queries, please don't forget to ask me in my Udemy inbox and things like that. Also, if you are going to install some data formats, also going to work with some data formats that I can speak right now. There are some bioinformatics data formats that you can also work with, like FASTI, BED, GTF, GFF, and R has powerful function for reading and writing the data in various format as well. So read table, read CSV. So CSV is also an Excel file, and we can also use R for accessing the bioinformatics data from online resources like the NCBI Geo database, Ensemble database, and we can also use their APIs and web servers. Some popular packages for using this is Biomarker, which we are also going to use for Ensemble, and that wraps up our whole module, I think so. So that was it for our whole introduction lecture. Thank you so much for joining for the module 1. In the next session we are going to deep dive into the data manipulation and visualization in R, and also going to perform some major analysis that are now being done in using the R bioinformatics technique. So thank you so much.